There are families in which cancer occurs more frequently than in the rest of the population. Indeed, in some cases, tumors have a genetic origin. This means that they are caused by the presence of a mutation that passes from generation to generation, increasing the risk of developing cancer for those that carry the mutation. The study of families with high risk of cancer has provided scientists with an opportunity to identify genes and mutations associated with increased cancer risk. The possibility to distinguish within the high-risk family between members that have a normal gene sequence and members that carry a high-risk cancer mutation is really important. The carriers of a normal gene have the same risk of developing cancer as the general population, while the carriers of a mutated gene have much higher risk of cancer. The identification of members that carry the mutation occurs through a genetic test. Starting from a blood sample, genomic DNA is purified and analyzed. If the mutation present in the family with high risk of cancer is already known, only a precise gene region is sequenced in order to verify the presence of the specific mutation. The genetic test is positive if the mutation is found. Family members positive for a genetic test will enter a specific cancer surveillance program in order to diagnose the potential development of the disease early on. The genetic test is negative if the DNA sequence is normal and the mutation is not present. Family members negative for a genetic test have the same risk of cancer as the general population. If in the family with high risk of cancer the mutation is unknown, a group of genes is analyzed in the genetic test in an attempt to identify the mutation. The genetic test is positive if a mutation associated with an increased risk of cancer is detected. In this case, the patient will enter a specific cancer surveillance and prevention programs, while relatives can perform the genetic test if they wish to. The genetic test is negative when no known mutation associated with an increased risk of cancer is found. The genetic test is uncertain when it identifies a mutation, the effect of which on cancer development is still unclear. In this case, based on the family history of cancer predisposition, clinicians will direct relatives toward cancer surveillance programs. The interpretation of data arising from genetic tests is complex, as well as the ethical implications for the patients and their relatives. That is why genetic tests are performed in specific hospitals with appropriate genetic counseling offered to patients and relatives. Discovery of mutations that lead to an increased risk of cancer has been important to develop specific genetic tests to identify members of the family that carry mutation before the onset of cancer.